so in their gut, the microflora that they have, there is evidence of some horizontal gene acquisition because they found genes from marine organisms in the gut microorganisms of the Japanese that allow digestion of this porphyrin. That's not quite the name. Yeah, porphyrin, carbohydrate that is in seaweed, which is otherwise indigestible. Which is kind of interesting that sushi is, the seaweed that's used to wrap the rice and the fish and so on is not naturally digestible. We don't have that enzyme. And I think, even the Japanese don't have that enzyme. But the Japanese and probably other people now have microorganisms in their gut that have acquired that gene that breaks down the porphyra seaweed. So just for interest in kind of environmental nutrition and maybe another example of horizontal gene acquisition from the marine microbe to the gut microbe. So that's for your information, interesting stuff. I'll announce, when am I going to have that study session? Thursday or Friday? Who wants it Thursday? Who wants it Friday?
Friday.
Okay, so everybody who wants it Thursday, raise their hands. Okay, we'll get an estimate. Okay, and then Friday. Oh man, it looks like the Fridays barely have it. So Friday at something like 4:30 till about 6. So I will announce that, hopefully over in Foster Hall. Okay. I don't have a bumper sticker. I don't have a parking sticker on my truck. I've got to remember after class to go get a parking sticker. I don't know what happened to it. I think it was stolen. And I've gotten so many parking tickets.
You can find the boot on it.
Oh, man. That's all I need is a boot. Okay. Here's then where we are going to go into some detail in how organisms strip electrons from organic food material and use it to make ATP. So chapter 13 was kind of introductory. I hope you understand that we're generating NADH, generating electron carriers, having stripped those electrons from glucose, and now we're going to see what microbes and man do with it. Because pretty much everything that microbes do with it, we do too. Okay, so chapter 14. So moving electrons. And I've tried to emphasize electrons, and then in this chapter, protons are really a more elementary energy source than ATP. You have to have electron and proton movement before you can ever have ATP. And so that's at a more basal level, more maybe important level, is to track electron and proton movement in order then to understand where ATP comes from. So electrons come from reduced food molecules, think glucose, onto energy carriers, think NADH, FADH2, and then onto membrane protein carriers, and then finally onto oxygen. So that's aerobic metabolism, but microorganisms can switch to anaerobic metabolism and use a whole bunch of other alternate electron acceptors like nitrate and chlorobenzene, iron and carbon tetrachloride. So in other words, microorganisms have a much more diverse ability to breathe compounds like iron, nitrate, carbon tetrachloride that we cannot do at all. Okay, so we've talked about the proton motive force. We're going to see how kind of classically it's generated. You should have had this material before. And so we are going to review that and then give it a little bit more detail. Okay, so the proton motive force, I call it the raw energy to make ATP. It's the protons and electron movement that really can facilitate ATP production. This was the cover of a journal, I don't know what journal, maybe it was Science, where we have scientists that have discovered enzymes in Rhodobacter palustris, which can generate hydrogen gas using sunlight. And so it's kind of cool. And if you can make hydrogen gas, you are now making potentially the energy source of the future. And so here's this picture of the environmental engineer who is capturing sunlight energy. And here is this, this is a gram-negative bacterium, kind of getting the royal treatment there. It's kind of a fun cartoon. Sipping a straw of nutrients from a lake, but making hydrogen. So you can see the gas line of hydrogen going to the car. So pretty cool picture in the front of your chapter 14. You know, neat metabolism. Metabolism can be used to make our own energy sources. But a few years ago, Dr. Condon, Deng, and myself got the patent on this. And so we have, and this is of course led by my engineering colleagues, developed a system where we have a light source right in here to stimulate those very kind of organisms to make hydrogen gas. And so this was a patent that was administered in 2012. And, you know, just a fun, privileged work to be able to work with engineers to get these patents. And we've continued to patent a couple of other things along the lines of making hydrogen gas from biological systems. Okay, so electron transport systems. So electron donors and electron acceptors. So glucose is an electron donor. Oxygen is an electron acceptor. So you can kind of start there with the big examples of what compounds are used as donors and acceptors. And we're going to be looking at how in biology you want a whole bunch of little steps in terms of energy change. You know, you don't want to have a firecracker going off in a cell releasing a lot of energy. You need to release energy in kind of small steps that are biologically compatible. So same thing with the electron transfer. It's a number of electron transfer paths are leading towards that ATP production. And typically, or it ends up being in a membrane where you basically have electricity. So we have a, I mean, I already talked about, we're going to talk about it at the end of this lecture. You can capture this electricity that is running through biological membranes and use it to run devices. Okay, in the electron transport system, and you can also see this abbreviated electron transport chain, a chain of proteins in that membrane. So those two are interchangeable, ETC and ETS, electron transport chain and system. Okay, so like I said, membranes are really important in terms of where those electrons are flowing and then making energy. And so electron transport systems are proteins that are embedded in a membrane that separates two aqueous environments. And you know, when you're talking about a membrane, you must be talking about a semi-permeable membrane, so that there's not free interchange from outside inside. There's this barrier, and because of that barrier, you can have gradients of protons so that you can make this proton motive force. So ion gradients, think proton gradients. More hydrogen ions on the outside than on the inside. Looks like a battery, and you can generate energy with a battery. Here's a couple of here's an electron micrograph on the left side, showing an organism that's got a whole series of membranes that then are colored with, these aren't exactly, this is a computer-generated image, but showing the layers of membranes inside the cell and how separation can occur in terms of the proton gradients that can form. And I think that's figure 14.3, not figure 143. Okay, so let's review what the proton motive force is and how it generates two, well, we're not quite there in terms of what it's generating. Okay, so transfer of protons through a series of proton pumps generates what we call an electrochemical gradient of protons called a proton motive force. And what does that PMF do? What does it do? It drives the conversion of ADP to ATP through this marvelous biological motor called ATP synthase. And so this process is known as the chemiosmotic theory. So, just briefly, from sugar metabolism, electrons are transferred to a membrane, and because of that electron transport in the membrane, protons are pumped and are used to drive ATP synthesis through the ATP synthase enzyme. So those two sentences will get you far in terms of describing how cells generate ATP from proton motive force. I just make a little point here. We're still calling this a chemiosmotic theory. It was developed in 1961, and it's really been established, and it's just sort of interesting how we still use theories, the word theory. We've just added evidence to it over the last 50 and 60 years, and I have this kind of pet theory that physicists are not afraid to make laws, and biologists are. So we're still clinging to this theory. I don't think it's a theory anymore. And we just need to say, we have a chemiosmotic law. This is what happens. You can isolate the enzymes, you document the protons. It's happening in algae, trees, and tigers. But we just don't have what it takes, man, to call things a law. And that's not true of all of our theories, but anyway, it's just kind of an interesting thing. The difference between all the laws of physics. How many laws of biology do we have? Like the cell theory, right? We're still calling that the cell theory. This is theoretical, that we're made of cells. No. It's a law. It's how biology is organized. Okay. Proton motive force. So we're going to, I hope this doesn't get too redundant, but here is then the separation of the two components of the proton motive force. You generate an electrical potential, so you have a charge gradient. You have a lot more positive charge on the outside of that membrane than on the inside. So when you have charge separation, you have a battery. And so that is an energy source just like a battery is. And then you have this chemical difference. So you have hydrogen ion. It's not just charge, it is a chemical gradient. So you have a lot more protons, a lot more hydrogen ions on the outside, forming a chemical gradient. So understand that there's a charge gradient and a chemical gradient that's formed when protons are pumped. So you can measure that chemical gradient with a pH meter. And you measure, on the inside of the cell, it's about pH 7, but the cell has pumped all these protons to the outside of the cell, and now there's like 100 times more protons on the outside than on the inside, and so it's a pH of 5. It's pretty acidic right outside that membrane. So here is kind of a diagram illustrating kind of the difference between charge and chemical gradients. So a charge gradient is just looking at positive ions versus a lot more positive charge outside than on the inside. And you can measure that. The delta P is then described by the Gibbs, that's not Gibbs free energy reaction, that's the Nernst equation. So this is a charge gradient and a pH gradient that's being shown in A, whereas in B, you have a charge gradient with no pH gradient. So you have, yes, the same charge difference, but in terms of the protons, you've got the same amount of hydrogen ions on the outside as on the inside. And then these are, like I say, just a way to help you see the difference between a charge and a chemical gradient. So here is a pH gradient with no charge gradient. So you've got a lot more hydrogen ions on the outside, but in the end, because of potassium ions and sodium ions, there is no charge gradient. So it's just kind of a helpful diagram to help you differentiate the difference between a pH gradient and a charge gradient. They're not necessarily the same thing. So typically, in a biological cell, both pH and charge gradients are common, and those and our membranes are charged. They are energized, is another term for that. There is this proton motive force pretty much continually on our membranes. And so this is typically the system that we're seeing in A, both the charge and pH gradient. And these are sort of learning tools. Okay, so we've talked already in this course about a number of the functions that require proton motive force, and this brings just a few of them together. A lot of symports, we've talked about the uptake, the simultaneous uptake of lactose with a proton. Well, that's using this proton motive force to take up lactose in this symport that we talked about in the very beginning of the course. So a lot of uptake mechanisms are driven by that proton motive force. We already know that flagellar motion is driven, that the fuel for that RPM that's generated with flagella is protons, not ATP. It's protons running through this biological motor. And those are the main functions that are shown in terms of what uses the charge gradient, because this shows you how that charge gradient is made, and that's basically through protons being pumped and then utilized by ATP synthase. In terms of electrons, you can kind of think of electrons